Welcome to The Explainer. Today, we're jumping right in to Chapter 8, the unit of life. We are embarking on a journey to answer literally one of the absolute greatest mysteries of our universe, the mystery of life itself. We're going to focus our lens right down on the fundamental unit of life, the cell. You know, whether you're looking at a microscopic organism or the person sitting right next to you, we are all tied together by this incredible microscopic architecture.
So here's our roadmap for this explainer. We'll start with the mystery of life, then look at formulating the cell theory. From there, we'll step inside the living arena, check out prokaryotes, rapid and simple, then eukaryotes, complex compartments, and wrap up with the ultimate cellular boundary. Let's get into it.
Section 1. The mystery of life. Look around you for a second. You see living things, and you see inanimate objects. But have you ever wondered what exactly separates the two? I mean, what does a living thing have that an inanimate object simply doesn't? Well, the answer, at its absolute core, is the presence of the basic unit of life, the cell. Anything less than a complete cellular structure just does not ensure independent living. It is the fundamental structural and functional unit of literally everything alive.
To truly understand how these cells work, scientists use a really fascinating approach called reductionist biology. Think about it like this. Gross physiological processes, things like digestion, excretion, or even memory and defense, are broken down into their molecular and chemical components. We're essentially asking, what are the molecules actually doing inside the cell to make these massive, large-scale processes happen?
And a true pioneer of this physicochemical approach was G.N. Ramachandran. Using the principles of physics and chemistry, he made some outstanding contributions to structural biology. Most famously, he discovered the triple helical structure of collagen way back in 1954. He's a perfect example of how analyzing life at the molecular level unlocks its greatest secrets.
Moving on to Section 2, formulating the cell theory.
So the discovery of the cell wasn't just a single aha moment, right? It was more like a collaborative detective story. Tracing the timeline, we start in 1838, when a German botanist named Matthias Schleiden examined plants and realized they were all made of different kinds of cells. Just a year later, in 1839, Theodor Schwann, a zoologist, reported that animal cells had this thin outer layer. Together, they realized both plants and animals were made of cells and cell products, but they couldn't explain how new cells formed. That crucial missing puzzle piece? It was finally placed by Rudolf Virchow in 1855.
Virchow summarized this with a catchphrase that became incredibly famous, omnis cellula a cellula, which simply translates to, all cells arise from pre-existing cells. Boom! He gave the cell theory its final shape. Today, we understand that all living organisms are composed of cells, and every single cell alive today came from a previous one. It's this incredible, unbroken chain of life.
Section 3. Inside the living arena.
Now, inside both simple bacteria and complex human cells, you're going to find the cytoplasm. It is quite literally a semi-fluid arena. This is the main stage of cellular activity. This is where the magic happens. Various chemical reactions are constantly occurring right here in the cytoplasm to keep the cell in a living state. Without this bustling arena, life just ceases.
Section 4. Prokaryotes, rapid and simple.
Okay, let's look at prokaryotic cells, which include bacteria, blue-green algae, and mycoplasma. Now, they completely lack membrane-bound organelles, and they're generally much smaller than our cells. But because of this simplicity, they multiply incredibly rapidly. They usually exhibit 4 basic shapes. You've got the rod-like bacillus, the spherical coccus, the comma-shaped vibrio, and the spiral spirillum. And despite lacking internal walls, they are highly adaptable. And even without complex organelles, prokaryotes have some brilliant, unique features. They store reserve materials right there in the cytoplasm, inside non-membrane-bound inclusion bodies. Many also have extracircular DNA called plasmids, which can actually give them unique traits, like resistance to antibiotics, which is a big deal. They also have mesosomes, which are specialized infoldings of the cell membrane. It's a minimalist setup, sure, but it is highly effective.
Section 5. Eukaryotes, complex compartments.
So while prokaryotes are kind of like a giant open-plan warehouse, eukaryotic cells, which make up plants, animals, fungi, and protists, feature extensive compartmentalization. They use membrane-bound organelles to basically create specialized rooms for different chemical reactions. They possess a highly organized membrane-bound nucleus to protect their genetic material, plus complex structures like the endoplasmic reticulum, Golgi complex, and mitochondria.
But even within the eukaryotic empire, there's significant diversity. When you compare them, plant cells are equipped with a rigid cell wall, plastids for things like photosynthesis, and a large central vacuole. Animal cells, they lack all of those. But on the flip side, animal cells have centrioles, which are absolutely crucial during cell division, and those are totally absent in plant cells. It's a perfect example of form following function.
Section 6. The ultimate cellular boundary.
Now, the detailed structure of the cell membrane was only fully deduced after the electron microscope was invented in the 1950s. It's made of a phospholipid bilayer, those blue heads and red tails, and it's studded with integral and peripheral proteins. You can even see bits of cholesterol tucked inside there. It's essentially a fluid, dynamic mosaic that wraps around the entire living arena of the cell.
So, why does this specific structural mosaic actually matter? Well, because it is a selectively permeable boundary. It doesn't just block everything out, it carefully facilitates the transport of specific molecules in and out of the cell. It maintains the exact internal environment required for all those complex chemical reactions to occur. It is the very structure that ensures independent living is even possible.
And that brings us to our final thought for this explainer. We've seen that through reductionist biology, everything we do, our memories, our defenses, our energy, is really just chemistry happening inside this highly organized microscopic unit. If all life basically boils down to the cellular level, I want to leave you with this. As our tools get better and we look even deeper into the cytoplasm and the nucleus, what incredible mysteries of life will the cell reveal to us next? Thanks so much for tuning in, and keep questioning the incredible world around you.